Hello everyone, welcome to my lecture on single radial immunodiffusion. I am Dr. SKM Habeeb, working as an associate professor at the SRM University in the Department of Genetic Engineering. So, the single radial immunodiffusion is a type of an experiment where we try to understand the presence of antigen and also its quantification. The principle is such that the interaction that is taking place between the antigen and the antibody is by means of a precipitation reaction. So, therefore, when an antigen and an antibody interact, they form visible cross-linking and they precipitate when the proper ratio of antigen to antibody is present. In this method, we make use of agar discs, and antigen and antibody are made to react in the wells. Also, specific antigens to the impregnated antibodies diffuse through the agar in all directions from the well and thereby react with the antibody present, forming visible precipitation ring. So let's have a look at the procedure. In this method, a glass slide is taken and agar is poured on that glass slide. Once the agar is polymerized, then we make cuts in a circular fashion on the agar slide, and then the antigen is loaded onto the agar, which already has the antibody added to it, and then the antigen diffuses radially, and thereby formation of a ring of precipitation will form in the area of optimal antigen and antibody concentration. So let's have a look at the demo of this experiment. Now this is 0.1 gram agarose is taken up and added to 10 ml phosphate buffer. So therefore, what we do now is take this solution and place it and ensure that it is heated up as the general procedure is. Once the agarose solution is heated and cooled, and it's the time now to go ahead and add 1 ml of the antibody to the agarose solution that was prepared. Ensure that the antibody is added not in the hot agarose solution, because this may lead to denaturation of the antibody. So extreme care must be taken that the temperature is appropriate enough for the antibodies to be added. So once the agarose has cooled, what we'll do now is we'll take a glass slide and we will pour the agarose solution that is consisting of the antibody in it onto this glass slide in a nice fashion, so that we get a uniform piece of a gel on the glass slide, which is acting as a matrix or as a support. Once the gel is polymerized, solidified, we will make use of a gel puncture apparatus and make wells by sucking up the gel from that disc. So this will lead to the formation of circular disc type wells, as you can see. So we have right now a gel which is consisting of the antibody and also the gel has numerous wells cut out, as can be seen from this video. We can mark down these wells that are being cut out as A, B, C or D for our reference and observations. Now you can see over here there is a clear formation of wells. Let's take test A and we will add that in one of the wells. Likewise, all the wells will be loaded with different types of antigens of different concentrations depending upon the experiment that we are going to perform over here. So we had two tests, test A and test B, and we are also going to add 4 standards in the form of standard A, standard B and standard C and standard D. As it's the routine procedure, these standards will help us in the determination of the test concentration T1 and T2. Now we will take this loaded gel and make use of a petri dish, keep a wet moisturous cloth, and on top of it we will place this in this fashion, and it will be incubated at room temperature for approximately 20 to 30 minutes. Let's do it. Once it is done, the glass slide is removed from the incubator and is witnessed under the dark surface, and from this you can see there is a nice formation of precipitation ring in a circular fashion. This is the reason the name of the experiment is single radial immunodiffusion. That is, the antigen diffuses through the gel in a circular fashion as there is antibody present in this gel as well. There can be seen in this glass slide that the diameter of these rings are of different lengths, because that defines the concentration of a specific standard or a specific test antigen. So therefore, the diameter of the precipitation ring is directly proportional to the concentration of the antigen present in our test sample. So we'll make use of these standard A, B, C, D, and therefore plot a graph, and from that we will be able to determine what is the concentration of the antigen, that is T1 and T2. So What do we see in the results over here is the presence of precipitin ring around the antigen wells indicates there is a specific antigen and antibody interaction has taken place. If the specific antibody is not there in the gel matrix, then obviously whatever antigen you may have added as the sample, it will not form a precipitin ring, and therefore that indicates there is no reaction taking place. Absence of precipitin ring suggests absence of the reaction. Greater the amount of antigen in the well, the farther the ring will form from the well. Let's have a look at the applications. Immunodiffusion techniques are mostly used in immunology to determine the quantity or concentration of an antigen in a given sample. As was detailedly explained in the previous slides, we can estimate the IgG, IgM antibodies in sera and also to detect the influenza viruses. The method can also be utilized to compare two different antigens to determine the purity of this antigen preparation, and also it will help us in the diagnosis of different diseases. With this, I hope you have been able to understand the single radial immunodiffusion method, and I thank the research scholars Bhavya, Kaushani and Srinitya for assisting me in the preparation of this video. Thank you. Catch you later on a different video. Have a good time.